How? Assuming that life evolved from organic molecules that formed on the Earth, how could this have occurred? The question is not novel. Charles Darwin wrote a letter to Joseph Hooker, dated the 1st of February, 1871, in which he made his famous reference to the life originating in a warm little pond. It is often said that all the conditions for the first production of a living organism are now present, which would ever have been present but if, and oh what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, and present that a protein compound was chemically formed, ready to undergo still more complex changes. At the present day, such matter would be instantly devoured or absorbed, which would not have been the case before living creatures were formed. End quote. Scientists disagree about how easy this process would have been. Biochemist Christian de Duve said, "The universe was pregnant with life. We belong to a universe of which life is a necessary component, not a freak manifestation." End quote. Others, however, point out the many difficulties of producing the first life forms. First, in producing the molecules themselves, then in assembling the complex organic systems in which these molecules function, then in the formation of the first cells. Producing the molecules. Biological molecules can form from inorganic molecules. Johannes Berzelius, one of the most famous chemists of the early 19th century, said in 1827 that organic molecules could not be made from inorganic sources. But the very next year, his friend and former student, Friedrich Wöhler, synthesized urea, known at that time only from the kidneys, simply by heating ammonium cyanate. Louis Pasteur showed that life comes only from life today and under normal conditions. It is the law of biogenesis, or life beginning. But many 19th and early 20th century scientists speculated about the abiotic origin of life. Again, abiogenesis, especially concerning these questions. Number one, what was the energy source? To make simple inorganic molecules into complex organic molecules, energy is necessary. The early Earth had numerous sources of energy for the synthesis of organic molecules, such as lighting, ultraviolet light, cosmic radiation, and heat from asteroid bombardment, as well as volcanoes, only to name a few. Second, what was the atmosphere like? During the mid-20th century, the atmosphere of the early Earth was assumed to be reducing, consisting of molecules such as methane, water vapor, hydrogen, ammonia. This was a reasonable assumption, as these molecules are common in the atmospheres of large gas planets such as Jupiter. By the late 20th century, however, most scientists believed that the early Earth atmosphere was neutral, which meant it consisted largely of carbon dioxide. Recent evidence suggests that the atmosphere might have contained a substantial amount of methane. Based on studies of minerals from the earliest sedimentary rocks, and it's called siderite, some scientists have suggested that the atmosphere could not have contained enough CO2 in order to have produced the greenhouse effect that was known to have prevailed on the Earth at that time. And that methane, a potent or strong greenhouse gas, might have largely been responsible for the warmth of the atmosphere and Earth. The only fact on which there is universal agreement, however, is that the atmosphere of the early Earth contained virtually no oxygen. And so, we come to question 3. Where did life originate on the Earth? Many scientists assume that this must have occurred in shallow seas, One problem with this hypothesis is that the same ultraviolet radiation that would provide energy to organic syntheses would also have destroyed the molecules as well. Some scientists, such as geologist John Corliss, have asserted that life originated in deep ocean vents where water meets lava. This has the disadvantage that the heat itself could have destroyed the organic molecules as easily as it created them. Some bacteria that live under those conditions today have special adaptations that prevent the heat from disrupting their molecules. The deep sea vents have abundant ferrous ions, which would have produced a reducing environment and encouraged the synthesis of organic molecules. Fourthly, what is the evidence for the origin of organic molecules on the early Earth? Many scientists, starting with Ivanovich Oparin in Russia and J.B.S. Haldane in England, have suggested that organic molecules came into existence under the condition of the primordial Earth. During the second half of the 20th century, numerous simulations of early Earth conditions have been conducted in laboratories. The earliest and still most famous of these simulations was conducted by biochemist Stanley Miller in the year 1953. And this is called the Stanley Miller experiment, in which he put a mixture of reducing gases, including methane and ammonia, into glassware that circulated the gases through water and passed an electric spark. Organic molecules accumulated in the flask. Rather than being an incomprehensible mixture of molecules, the product was dominated by a few molecules: formic acid, glycine, glycolic acid, alanine, lactic acid, propionic acid, acetic acid. In much smaller quantities were urea, aspartic acid, and glutamic acid. 4 of these are amino acids. In 1960, chemist John Oro conducted a similar experiment that produced adenine. All of these molecules are used by organisms today. Amino acids are the building blocks of proteins, which are largely responsible for the complexity of life processes. The popular press got the impression that Miller had made life in the laboratory, a claim that he indeed never made. The world was astounded that the building blocks of life could be so easily produced by simple chemical reactions. Many other simulations have been performed since Miller's original experiment. Again, Stanley Miller. And his experiment was not the first synthesis of its kind. In 1913, Walther Loeb produced amino acids from wet formaldehyde plus an electric discharge. Loeb was simply studying the chemistry rather than investigating the origin of life. Scientists are a long way from figuring out how life might have arisen by spontaneous chemical reactions on an inorganic Earth. Fifthly, producing all of the necessary molecules. Although many organic molecules can be formed in the laboratory simulations, many of the molecules that are essential to life today have not been formed in these simulations. Ribose, which is a component of RNA, would have been especially difficult to form. In addition to these molecules, the simulation experiments have also produced a tar-like goo that covers the inside of surfaces of the flasks, and as biochemist Stanley Miller and Antonio Lazcano say, the primordial broth, they say, quote, may have been a chemical wonderland, but it could not have included all the compounds and molecular structures found in the simplest of present-day microbes, end quote. A particular problem has been in designing a one-spot reaction in which the scientist puts inorganic chemicals in and gets living cells out. In order to obtain the different molecules necessary for life, very different conditions of acidity and temperature are needed as well. For example, freezing conditions are necessary to preserve adenine and guanine, while for cytosine and uracil, warm evaporative conditions are needed. Biologist Robert Shapiro has been particularly eager to point out how implausible these prebiotic syntheses would have been, requiring an unbelievable configuration of heat and cold, salinity and purity, acidity and neutrality, a series of unlikely events occurring in just the right order to produce even a few of the chemical building blocks of life. The chemical simulations are, according to Simon Conway Morris, quote, highly artificial if not contrived, end quote. The outcome of the syntheses depends strongly on the starting conditions. The reactions would not work at all in a neutral atmosphere rather than a reducing atmosphere. Stanley Miller, for example, used a continuous spark plug, but when brief, stronger blasts of electricity, which stimulate lightning, were used, the results were disappointing. When the order of the glassware components changed, the results were different. Some experiments would not have worked without doubly purified water available in chemical laboratories, but not on the primitive Earth. So far, there has been little promise from one-pot reactions in explaining the origin of all the chemicals of life converging in one place at one time. Sixthly, origin of handedness in biological molecules. Organic molecules can have left versus right-handed mirror images, exactly the way that human left and right hands are identical but opposite to one another. Same thing about the brain, the hemispheres of the brain. Organic molecules are produced by non-living processes, and they are almost always chiral, which is an equal mixture of the two forms. However, proteins in organisms use left-handed amino acids and right-handed nucleic acids. A protein made of all left-handed or all right-handed amino acids is more stable than a protein made of a mixture. But how did the preponderance of left-handed amino acids ever get started in the first place? There was great excitement when it was discovered that meteorites could have an excess of left-handed amino acids, but this excess was only 7 to 10%. As it has been suggested that ultraviolet light polarized the amino acids before the meteorites reached the Earth. It is still unclear, however, whether this could have happened on the primordial Earth, and it has been demonstrated that divalent cations such as the calcium ion can produce an uneven mixture of the two forms of organic molecules. Seventhly and lastly, we will talk about the synthesis of large molecules from small precursors. The production of small organic molecules may have been easy on the primordial Earth, just as in nebulae and comets. But how could small molecules have assembled into large ones, such as the proteins and nucleic acids needed by modern cells? Large molecules tend to dissociate into smaller ones in water. There are two processes that may have allowed the formation of large molecules on the primordial Earth, and they are as follows. Activated precursors. Regular amino acids do not polymerize into proteins in water very well, but amino acids in the amide form do. As biochemist James Ferris explains, carboxyanhydrides can form into protein-like molecules in water. When this happens, he points out the resulting protein-like molecules form only the correct type of peptide bond that characterize biological proteins, and moreover, the resulting molecules up to 10 amino acids in length contain only left-handed components. Adsorption on mineral surfaces. Small molecules bumping into one another in a watery swirl would be unlikely to form the complex molecules characteristic of life. In many chemical reactions, solid surfaces allow the orderly catalysis of reactions. The catalytic converter of an automobile uses surfaces of the metal palladium to catalyze the reaction that eliminates carbon monoxide from the auto exhaust. J.D. Bernal, a British biochemist who's wrote several very good texts that I recommend, first proposed the possibility that the synthesis of large molecules from small ones may have occurred on the clay mineral surfaces. Clays certainly provide an enormous amount of surface area for such reactions to take place. Leslie Oriel calls this possibility life on the rocks and has demonstrated that RNA molecules of length up to 40 bases can be produced on a mineral surface. RNA molecules of this size would be long enough to get the RNA world started. All right. Now we'll talk about RNA world. Assembly of complex chemical systems. Modern life, even of the simplest bacteria, is too complex to have arisen directly. In particular, the elegant genetic system of DNA, the raw material of evolution, must represent an advanced system not found in the first life forms. In modern cells, DNA stores genetic information in which it is transcribed into RNA, which directs the formation of proteins, as microbiologist Carl Woese proposed in the year 1967. Scientists realized that a simpler genetic system of life must have preceded that of DNA in the modern cell. Once modern DNA cells came into existence, they would have outcompeted the more primitive form, which therefore no longer exists. Some suggestions that are not all mutually exclusive include some scientists, such as Stuart Kauffman, Gunther Wachter-Hasser, and Christian de Duve, have proposed that metabolic systems such as glycolysis, which releases energy from sugar so that organisms can use the energy to operate. And again, we're talking about this suggestion, and it has met most skepticism among scientists because at some point a genetic system would have to take over the control of the cell. The first genetic system may have been formed of clay mineral crystals rather than that of organic molecules, and this scenario proposed by British scientist Graham Karen Smith. Organic molecules such as RNA helped the mineral crystals in their replication. Later, the minerals served as scaffolding for the reactions of the organic molecules, which later took place without the scaffolding. The first genetic molecule may have been something like TNA or thionucleic acid or PNA, peptide nucleic acid. The formation of such molecules could have occurred more readily on the early Earth. In particular, TNA contains no ribose. It works in a fashion similar to DNA and RNA, and it can bind with DNA and RNA, which means that it could have transferred its genetic information to RNA during the evolution of a new RNA-based life form. In the year 2000, Israeli scientists proposed that the first genetic system may have consisted of lipid-like molecules that can not only form cell-like structures that grow, but also pass information into the new cells. It is widely accepted, following the lead of biochemist Manfred Ingen, that an RNA-based genetic system could have produced a DNA-based system for several reasons. First, modern cells make DNA bases out of RNA bases. Second, all modern cells use modified ribonucleotides as the basis for some of their essential metabolic chemistry, such as ATP and NADH, and some modern enzymes need small RNA molecules in order to help them carry out their reactions. Third, biochemists Thomas Cech and Sidney Altman, who won the Nobel Prize for Chemistry in the year 1989, showed that RNA can act as an enzyme. They are called ribozymes, and that is, RNA can be both genotype and phenotype. Some organisms, such as the protist Tetrahymena thomophilia, use ribozymes. Many scientists consider the ribosome, which is built of both RNA and protein, and in which it is the RNA that has catalytic activity, to be a remnant of the time when RNA was the blueprint of life, and in the laboratory, RNA can catalyze its own reproduction. Strings of RNA containing guanine result when RNA molecules containing cytosine are used as a template. Leslie Orgel calls this the molecular biologist's dream. More than 20 different ribozymes have been produced from random RNA mixtures following by a selection experiment, including a ribozyme that synthesizes a nucleotide from a base and another ribozyme that makes more RNA. And so here we say that it could be that RNA-based genetic system would have preceded a DNA-based system, and that DNA comes from RNA. Chemist Walter Gilbert proposed the RNA world scenario in the year 1986. In this scenario, the primordial seas were filled with RNA molecules that replicated themselves and therefore constituted a primitive form of life. Later, according to evolutionary biologist John Maynard Smith, and these RNA molecules were assisted by amino acids, which resulted in the origin of the genetic code. Still later, complex DNA replaced simple RNA as a more stable form of genetic information, but living cells never got rid of the RNA completely. This process could or would necessarily occur more efficiently if the RNA molecules consisted of bases that were all left or right-handed, which could explain the origin of handedness in the genetic molecules. In RNA selection experiments, smaller RNA molecules replicate faster than large ones, which makes it difficult to explain how complex molecules would have arisen in an RNA world. And so the last universal common ancestor, LUCA, or LUCA, of all cells, and so instead of Lucy, it's the last universal common ancestor of all cells that are alive today, stored genetic information in DNA. But the original life form may have used RNA or an even earlier chemical basis that we just don't know about today. So, what about the formation of the first cells? Well, the final step in the origin of life would be to explain how the life reactions would have been isolated into cells, a step necessary to keep the waves of the ocean from separating and diluting them. Most modern cell membranes are made from phospholipids, which are molecules that can bridge the gap between fatty and watery molecules. Lipid-like molecules are today found in sea foam. Some scientists suggest that this sea foam in shallow primordial ponds may have formed the first cell membranes. Other scientists have formed micelles, which are clusters of molecules that carry out chemical reactions and replicate themselves in the laboratory, and they propose that the first cells may have resembled these. At some point, the origin of cells must be explained, and the protocell. May life have been in operation for a long time before it was compartmentalized into cells, according to Carl Woese, who has described a life state that preceded life forms. In conclusion, scientific research has illuminated many possibilities for the origin of life, in particular, answers to the questions of when and where. But for now, scientists will have to be satisfied with not knowing a definite answer as to the questions of how. Now, and so we've discovered what, we've discovered when, where, and how. Scientists normally focus on the how. How do things happen? How do things operate? How do things function? How do we do this? But also, in philosophy, we have how do we know in epistemology. How do we know that something's true or not in history? How do we, and so history is more in line of discovering, the what, when, where, and why. What, when, where, why. But history also tries to explain the how. How did something happen? Just like science is the study of the behavior of things, so is history the study and behavior of particularly humans. History is really the study of human behavior, but also psychologists like to study those sorts of things as well. So that's our reading on the origin of life. We don't know where life came from, but we might find the answers to our questions surrounding the origin of life on our planet.